The International Festival of Arts and Ideas is created and produced on the traditional lands of the Mohegan, Mashantucket Pequot, Eastern Pequot, Scotticoke, Golden Hill Paugussett, Niantic, and the Quinnipiac, in the land we all call home, Connecticut. We hope that from wherever you are, you take a moment to acknowledge and honor the native people whose land you are on and the history of the place you are in.
The International Festival of Arts and Ideas stands in solidarity with Black and Brown communities and other groups who are targeted and abused by unjust systems of oppression. We support all movements working to decenter white voices and dismantle white supremacy. We are actively seeking to dismantle systemic racism. We raise our voices with those in our community who are already engaged in this vital work. We commit to working alongside you to create transformative change in New Haven and in our local community.
Good evening, everyone, and thank you for coming out to the Yale Repertory Theater. And a big thank you to those who are watching virtually, the virtual stage at NBC Telemundo. This is the 27th year of the festival, and I'm very honored to be the curator of the Ideas program this year. My name is Denise Santisteban. Before we get started, I'd like to thank the Connecticut Humanities, whose partnership makes the Ideas programs possible and ensures that we can do over a dozen Ideas talks, all for free. And a thank you to our marquee sponsors who help us keep things rolling: the Community Foundation for Greater New Haven, Yale University, National Endowment for the Arts, Connecticut Office of the Arts, City of New Haven, and once again, Connecticut Humanities. I'd also like to thank the Ely Center of Contemporary Art, who has made this talk possible. With their partnership, we're very appreciative that we could bring this person to speak with you today. Make sure you check out all of our programming at artidea.org. And I want to give a shout out to our Ideas program that's going to be here tomorrow evening at 5:30, Sean Taylor, The Power of Fandom. He's a dynamic speaker who'll be talking about how we can use fandom to change things for the greater good. So I hope you'll join us again tomorrow evening. And now it's my great pleasure to tell you about our speaker for tonight. Her name is Suzanne Anker. She's a pioneering bioartist whose work is bridging art and science. She founded the BioArts Lab School of Arts in New York City. I first learned about her work through Debbie Hess at the Ely Center. And she told me she first met Suzanne in 2000 when she was part of the art space show Untitled Spaces. And it was just such incredible work that she knew that she had to come back again. So we're honored here to have her talk to you again 22 years later. And I want you to know this is in partnership with the Ely Center, and after this talk, there's a reception, and you can see the exhibit After Eden, which is showcasing for the first time some of her newest work. So please join us at the Ely Center after this talk, 51 Trumbull. And one other thing, on the back of your programs is a QR code. So as you watch and listen, if you have a question, you can just scan that code, type in your question, and we'll be able to present it to Susan afterwards. And thank you very much for coming out again, and it is my great pleasure to introduce you to Suzanne Anker. Suzanne? Thank you.
Well, welcome everyone, and thanks for coming out this evening. And I would like to introduce you to the concept of bioart, which you probably don't know what it is, because basically it's a very hard concept to get your mind across. And I'll be showing you some of my work, and some of this work is at the Center for Contemporary Art, where you could see it in person. And let's now begin.
A new generation of biohackers is tinkering with DNA to create plants with new colors. At the School of Visual Arts in Manhattan, plant researcher Sebastian Cachobo is trying to create genetically modified plants, mostly from information he learned on the internet. One of his pursuits in the school's bioart lab? The blue rose, considered the holy grail of horticulture.
I think the main reason why people are trying to make blue plants is because there are very few, if not any, blue plants out there that are manipulatable or growable in a specific condition. Basically because those high saturation colors just simply do not exist in nature.
GMOs, or genetically modified organisms, have drawn a considerable amount of controversy, with concerns about the potential impact on health and the environment. Cachobo doesn't think such worries apply to projects like his.
The main argument I would have towards anybody who would contest us going through with this project is the fact that it'll be contained, and it's a plant. So we're not working with pathogens or any other type of human or mammalian tissue.
Suzanne Anker sees the process as more of an art project than a science experiment.
Artists here use science as a tool, so they are not engaged in research in the same sense that a scientist would be. Blue is kind of an area of wonder. Children say, why is the sky blue? All the time. That's one of their first questions. And I think that is in part the background of this. But I do think that even if it doesn't work, it doesn't matter because it's art.
The process is meticulous and complex. It all comes down to identifying and manipulating a specific gene that could give the plant its color.
The gene in question, called AE blue, produces a beautiful cerulean hue in bacteria. It's never been tested in plants as far as I know. So this will be the first time that we actually try to put it into a plant.
With the project about halfway done, Cachobo is still tackling problems, but he says even if it fails, the experiment won't be a loss.
In an ideal situation, in another 3 months, we'll have our blue plant. Worst case, it might not work at all, and that's the risk that we're willing to take, and that's the risk that we want to attempt, because even a failure might be useful in terms of understanding the processes at hand.
All right, well, this raises several questions. One is, why would the Wall Street Journal put this on the front page of their newspaper? This is a bioart lab in an art school. That never happens. And the second thing is when I said, if this doesn't work, it doesn't matter because it's art, that was not a snide remark. What I mean by that is that for every kind of scientific innovation, there is a counterpoint in the cultural sphere that deals with a very similar concept. So the concept of a blue rose goes back to the 18th century, to the poet-philosopher Novalis, who wrote about this love affair that he had had, and the blue rose became a symbol of the ephemeral, of infinity, of the eternal, and all of those things that are almost impossible to achieve. But again, the blue rose would appear in Stanley Kubrick's Eyes Wide Shut, when Nicole Kidman is dancing with her partner who has a lapel of a blue flower, and it appears in David Lynch's films, and it appears in Walter Benjamin's writings. So it is a cultural icon. And scientists have been trying to make a blue rose for quite a while. In 1991, they started to genetically modify a pigment from a petunia and tried putting it in a rose, but that did not work, okay? All you could get was something slightly lavender. So, like Novalis, in which this idea of the ephemeral, the infinite, the blue rose still stands today, both culturally and scientifically, as a magical kind of symbol. Now, what is bioart? Bioart is a tripart system. A lot of people contest what it might be. It's not amateur science, and it is really a kind of crossover or hybrid between the natural world and the cultural world. And there are three subsets that I'd like to introduce you to. One is by Mark Quinn, where you can kind of see this portrait of Sir John Sulston, who was a very famous geneticist in the UK, and he was able to convince Sir John to give him some cells, which he then duplicated and put in this stainless steel frame, so that when you look into this frame, you see yourself. And this piece now is in the National Portrait Gallery in the UK, the first time there is a portrait that is abstract. So we have wetware, we have the cells themselves. A second category has to do with the apparatuses that have been used in order to decipher DNA, for example, or other kinds of complex workings of the body, in which computer technology plays a significant role. So zeros and ones now can be used to create objects, and in this piece by me, which I'll talk about a little later, what we have here is a computer-generated sculpture, a sculpture I made without using my hands. Another very strange situation. And then finally, we have Patricia Picanini, whose work deals with more traditional sculpture, but not with traditional ideas. Here, excuse me, she has a cross between a human and a dog, and it kind of raises these questions about genetic engineering as how human does something have to be, or what are the rights of creatures that are like us but not exactly like us. And we had this conversation already about gorillas, and now this conversation is with genetically modified organisms. It's my first time speaking at a repertory theater. I've spoken at an abandoned rice silo, at a church, at a scientific lab, etc. And this was a piece I did at Cathedral of St. John the Divine in New York City. And it was a piece that used red and blue LED lights in order to grow vegetables in this cathedral. And lots of people came in to see this, because this church, this cathedral actually is world-renowned. It's the only Gothic cathedral in New York City. And some of the people coming in did not believe that this was real, that they had never seen plants in this color. And there were many conversations with the priests, as well as school children, etc. And the idea behind this piece really is twofold. One, because of climate change, we are now moving agriculture indoors. And if you would shine a light on some of the vegetables growing, you would see that they are actually green and not fuchsia. So fuchsia is the new green. Now, this idea was explored by NASA. And here you have a number of astronauts who are growing space lettuce, and I grew the space lettuce in the church as well. And because we are now starting to explore other planets for food sources, for precious minerals, etc. And what was really interesting about growing these for the astronauts was that botany in anti-gravitational conditions is nothing like it is on Earth. That when you plant a seed on Earth, you get one sprout. When you plant a seed in outer space, you get 2 sprouts. If you boil water in outer space, you get one big bubble. If you do it on Earth, you get lots of little ones. So this idea now of exploring galactically what can be made in alternative environments. Another set of work here is called Vanitas in a Petri dish. And for those of you who are familiar with art history, Vanitas was essentially a Dutch still life genre concept. And its idea behind it, during the Dutch Golden Age, where people were getting rich, and religious ideas were clashing with each other, the Vanitas paintings represented the wake-up call that materialism is not the only thing that one sort of makes a good life, but that there's a spiritual component to life as well, because everyone has an expiration date. So this was a moral warning. And in these pieces, what I've done is create something from nature and something from culture and blended them together in Petri dishes. So it's a reference also to this trajectory between what we make and what is natural, and that is the conditions under which we are living. We're living in a second nature. And here you can see that it also has a reference to synthetic biology, a piece of metal, a mushroom, some plastic, etc., to create this image. Now, the Petri dish is also a cultural icon because everyone knows what a Petri dish is. And Jules Petri, who was the assistant to Robert Koch, wrote one 300-word paper during his entire career as a scientist and discovered the Petri dish, which is used today. So for one 300-word paper, one gets to be famous. And the other thing about the Petri dish, from a cultural point of view, it's the place where hidden entities emerge from. So it is used as a metaphor as well as a scientific instrument. Here we can see some close-ups of the Vanitas pieces, a sunflower and a rubber ducky on the left, and a flower and some ceramics melded together here. And what I did after I made all of these Vanitas prints is I tried to see what would happen if I made them in 3 dimensions. And you can see on the left-hand side that these are the flat prints, and by creating a 3rd axis and using rapid prototype machinery, I was able to create these, which turn now from a still life to a landscape, and both of which are art historical genres. Here you can see a larger version of this. This was at a recent show at the Everson Museum in Syracuse, which was I.M. Pei's first museum in America. And for each Vanitas, one sees a rapid prototype sculpture. So what the visitors would try to do is match up the Vanitas with the remote sensing pieces to essentially see if they could figure out if they could recognize something that moves from 2D to 3D. Now again, remote sensing is another word from NASA. It's about surveillance data. It's about all of the pictures that we get through zeros and ones as these satellites fly around. And it is used for places that are either too toxic or war-torn in order to visit. So again, technology is biology, and biology is technology. And what interested me in these pieces as well is the images that were reflected through the glass. And some of the first, before the microscope was invented, glass beakers or jars were used as reflective devices to enlarge or diminish what was being looked at. So here there's all of these new kinds of images that are created. I want to say one more thing about this kind of work that, of course, the machine made. And something like this is nothing I would have done on my own. The machine read the data and decided that it was a collaborator with me and taught me about edges of sculpture that can be manipulated in new ways. So AI, which is this big program right now, not only do we teach the machine, but the machine teaches us. And here we can look at the close-up of this little world that has been now created with an X, Y, and Z axis. And it is a protrusion where you can set the field to how high or low you might want it. And something like a cookie here now becomes a crater. So the back and forth between 2D and 3D I find to be very enlightening. And here we can see the flower that I had mentioned before in 3 dimensions. Now there's this new body of work that I've been working on for the last 2 years. And as an artist, I think that one goes through different stages when one is developing a body of work, from infancy to childhood to adolescence to adulthood, etc. And during the COVID quarantine, I was working in my studio, kind of removed from the outside world, but still teaching my classes and running the fine arts department at SVA. But the project that I came up with was called After Eden. Now, Eden, of course, is a loaded concept for many reasons, and there are many interpretations of Eden. But everything in this series I grew myself. And through the 20th century's most radical sensibility, which is collage, which is cut and paste, which essentially displaces time and space and creates a new form. So from an ontological point of view, image A acts on image B to create image C that is nothing like image A or B. This kind of technology is like, in terms of art, is like genetic engineering, with the new types of programs such as CRISPR to delete or add genes. What we have, again, is cut and paste. So this becomes, for me, a way to use nature as the source to create new kinds of forms, because some are disappearing and some continue to evolve. And in this particular piece, we see this conglomeration of a flower that is also kind of like a bird. So what are the next set of images that we are going to see in the natural world? With the decline of pollinators and the ozone layer and permafrost, life is going to be very different very soon. The idea of growing strawberries in Greenland and cabbage in Greenland and mining for materials in Greenland after the snow has melted is going to create an entire new ecosystem. And here you can see, again, this cut and paste technique in which sort of plants get together and become something else. The image in the background here, which is out of focus, is also an ode to the disappearance of plants as we know them, as they become extinct. This one is very haunting to me because of its disjunction, and to kind of see the sexual organs of a flower in a different time zone with that of a spore of a dandelion. What we have in terms of evolution now through technology is fast forward. And I wonder what Darwin would have to say about this if he was alive. And here is another piece of mine with this process, cut and pasting these different types of plants together to create new ones. And I'm still working on this project. And over the last 2 years, what I have realized is I am no longer looking at the plant as a plant. I am now looking at it as a series of colors and forms, and they are becoming more and more abstract. And in this work, it appears as if it is a tropical scene, but you wouldn't have this kind of evergreen in the tropics. So the seasons are all mixed up. And I was thinking the other day, why don't we see pictures of Eden with snow? Now, if you read Genesis, it talks about the change of seasons, but we never see snow in the Garden of Eden. But in After Eden, we will see snow, and we may see a different season every day as we wake up and decide what we're going to wear that day. And finally here, one can see these as large scale. They have a very painterly presence and the geometric component that is manifested really is contrasted with the organicism that is evident in plants. And finally is my BioArt lab. Okay, this is one of the first BioArt labs in America, and it is a place where students use the tools of science to make art. And what's interesting about this is that when we invite scientists to come into our lab to teach us something or to show us how to extract DNA, the scientists are very scared of this lab because it's a cross between a 19th century lab and a 21st century lab. We have, sorry, I'm going to go back. We have plants, we have fish, we have coral. We just got a new pet, which is, we named Louise Bourgeois. We have a tarantula now in the lab because one of our students wanted to work with insects. And when scientists come in, they're not used to seeing an ecology in a lab like it was in the 19th century. But because scientists are very wary of letting artists into their labs, which I don't blame them, since artists don't generally pay attention to the rules, that it was necessary to make our own lab and work the other way. We have entered a number of competitions that we have scored very highly in, such as the Biodesign Challenge in 2016, where we genetically engineered a tomato to have a protein. This is before the Impossible Burger and all of that. And it was a speculative project, and we were able to get myoglobin into the tomato plant. We also did another project called Soiled, in which we tested 50 soil samples in people's backyards for the different nutrients. And that was at iGEM, and we won a gold medal for that project. So this has been a very interesting experience for me, kind of bringing together my interest in science and in art and in the wonder of the world. Thank you. Well, I think we have some time for questions.
Questions, turn over your program, QR code, and type away. I guess one of my first questions is, have they made that blue rose yet?
No, they've never made a blue rose that's blue. No, they've tried. They have made purple carnations,
Yeah.
And they have made other attempts to create the blue rose, but whoever makes it will be a billionaire, okay? Because blue is very rare in terms of the natural world. There are some blue flowers, of course, and there are some blueberries, but the natural world has very little blue. And part of that is because of pH factors that go into the coloration of fruits and vegetables and plants. And blue is very elusive which goes back to Novalis in being the infinite.
So those blue orchids that I see in Home Depot, those are not natural, those are,
No, those are dyed.
okay.
And you can dye a rose. You can take a white rose and put it into water with pigment, and it will come up through its circulation system. But it's like the seeds on Amazon that say, you know, these are seeds for blue roses. They don't work. I tried them. And other flowers that you see that are blue, many of them are spray painted. The only different one, and it's not quite cobalt blue, but it's close, is hydrangea. And if you have a hydrangea plant and you pour beer on it in the roots, it will change its color because it changes the pH factor that produces the pigment. So that's a very interesting kind of way to play in your garden.
We do have a question. When did bioart begin, and how has it evolved?
When did it begin? Well, it's hard to say. I would say in the 1930s, it wasn't called bioart, but Alfred Steichen, who was a very important photographer and also worked at the Museum of Modern Art in the photography department, genetically modified his delphiniums by making their single nucleus into a diploid nucleus, meaning instead of 2 chromosomes, it would have 4. And then what would happen when he planted these, they would become huge. And he's looked upon as sort of the father of bioart because these flowers were actually shown at the Museum of Modern Art. Okay, let's cut a couple of decades up to about the late 1980s, and the ideas around art and bio were now starting to brew. And there was a lot of advertisements that referenced DNA, which caught people's attention, such as if you drive a Volvo, you know, they use DNA as a safety device to drive a Volvo. Huh. And then in the year 2000, when the first draft of the Human Genome Project existed, there were many, many shows that brought together artists from all over the world, and conferences and articles, etc. So I would say the late 80s were the pioneers, and then picked up steam during the 90s, and now there's even a master's degree in bioart at St. Martin's in London. And it is an international community. It is not media specific, as I explained earlier. You could be a painter and be considered a quote unquote bioartist, because what happens at the end of the day is you enter into a philosophical conversation, that it all turns towards ethical dilemmas, which does not happen in a painting class or a drawing class, but happens in a bioart class.
That's a great segue to the next question. Are there ideas that have been deemed too risky by SVA? Are there any examples of something like that?
Other places like SVA?
No, ideas that were deemed too risky by SVA.
Yes, of course. We are a bioart lab level 1. There are many different levels, and in many movies you see, you know, when there's this big virus that has escaped and the scientists are wearing these jumpsuits and have no flesh exposed. We are a level 1 lab, which means that we don't deal with human cells, we don't deal with blood, we don't deal with HIV, we don't deal with other kinds of dangerous viruses. Because even working on some of the projects using biomaterials, you can create mold that could be very dangerous to you. There was a photographer who went around the New York City subway and took swabs with him and had the swabs put in Petri dishes and sent off to a lab to see what would emerge. And E. coli, Salmonella, Streptococcus, essentially all of those microbes are right out there in your environment. Even if I had a Petri dish now with agar and I left it uncovered in this theater and came back in a week, you would see things growing in it. So the question of safety and health is also very important in working with any kind of living material.
There's a comment about the After Eden series, that it seems to explore grief about loss of plant life, ecosystems as we know them, but also a sense of hope for ecosystems. Would you describe your personal feelings about climate change, about how climate change plays into your process for the work?
That's a great question. That's a great question. I am an optimist, and the sense of wonder I have when I see living things metamorphose is extraordinary. I mean, as a child, I used to go to the empty lot in back of my apartment building in Brooklyn and collect caterpillars and bring them home, and they would eventually form a cocoon and hatch. And I was just so amazed that a creature could go from a crawler to a flyer that I could never get that out of my mind. And I think that in a very good lecture by Margaret Atwood called Burning Questions, and she was interviewed by Judy Blume, and she was asked whether she was an optimist or a pessimist, and she said she was a soggy optimist, okay? Which was a great answer. I think humanity is resilient, and I think nature is resilient, and I think we've all become much more aware of the vulnerability that we possess, like expressed in the 17th century Vanitas paintings due to COVID, okay? This summer in my bioart residency, I had more students than I've ever had. And between climate change, sustainability, invasive plants, extinction, on one side, there's also new technologies like the COVID vaccine, which never could have happened without the decoding of the human genome. So there is hope, but the kind of hope I think was summed up best by this German philosopher who talked about the fact that we now are not thinking anymore in terms of utopia, that we've had that idea come and go for centuries, and he is offering utopia in miniature, meaning the individual and what the individual can do one seed at a time. So I think that there are no doors in the global world, and what happens in one space and time happens around the world. And this is something that we need to reassess about how we live under these circumstances.
That's wonderful. We have time for about one more question.
Sure.
So at the beginning you said that if the experiment failed, it didn't matter because it was art. Can you elaborate on that just a little bit more?
Yes. That if it doesn't work, it doesn't matter because it's art.
Right.
Okay. I think art and artists and creative scientists recognize accident in their work. And an example of this is Alexander Fleming, who was known to have a really dirty studio, okay? He put his Petri dishes with Streptococcus in his sink. They were open, and he left the window open, and air blew in. He went away for the weekend. When he came back, he saw that there was a fungus growing around some of these streptococci, hence penicillin. Another example is vulcanized rubber, where someone was able to figure out by accident that if you take a piece of rubber and you throw it on a furnace, it changes its consistency. Mirror neurons was another accident. And the relationship between creative thinking and innovation is very close, because I would call them both epistemological systems in which one deals with experiment that has no predictable outcome. And art is certainly like that. When an artist goes into their studio, et cetera, or is trying to solve a problem, they may try this, try that, try this, try that, and have an intuitive feeling that this can be done, but they don't know how to do it yet. So what I'm saying here is that this relationship between the cultural components that are reflected by science and technology is a huge backdrop. The kinds of paints that were made during the Impressionist times were synthetic, and therefore artists could have full access to a full palette. Cubism as well was a reflection of some of theories of relativity where people were thinking about this. Surrealism and its relationship to Freud. So if you go back into history, you will see correspondences. And that's sort of what happened to me in 1993 when I teamed up with a sociologist of science named Dorothy Nelkin, and we wrote a book called The Molecular Case, Art in the Genetic Age. Unfortunately, she died before the book came out, but it was published by Cold Spring Harbor Laboratory Press, a scientific organization of august renown. It's where James Watson is. And they published this as an art book. So, because what was happening scientifically was now happening culturally in terms of the understanding of genetics. And I think that DNA was a major influence on a lot of artists working in this area, because DNA is your bioarchive, and you don't even own your own DNA, okay? But that's for another lecture, okay? So I mean, what, you know, doesn't matter because it's art, because artists expect that a lot of what they do is not going to work out. Artists are great risk takers, and if it works out, it works out. If not, something else will work out. And I think that drawing these kinds of parallels between reason and intersubjective experience is what makes bioart so powerful.
Thank you. And thank you, Susan, for a riveting talk. Thank you very, very much. I know some of you have more questions, which means you just need to go to the Ely Center where Susan will be, and you can ask her there. But
Great.
Susan, thank you very much for coming.
Thank you.